So the honor of closing out the conference goes to my young colleague, Slava Epelman, who has built a team with collaborators Tanya Watts and Boris Hintz at the University of Toronto. So Slava is going to tell us about his successfully funded innovation project, Multifactorial Immune Cell-Based Development of Non-Ischemic Cardiomyopathy, which I think follows nicely from Gillian's talk.
Thank you, Mansour, for the introduction, and it's really an honor to be here. I can tell you for sure that this project wouldn't be moving forward without funding from the Ted Rogers. And also thank you for calling me young. And thank Gillian for introducing what is going to be a relatively complicated proposal. There's just no way around it. So I'm just going to dive right in. Heart failure. We've heard a lot about heart failure, but what I want to say is that the development of heart failure is modulated by the immune system. I think this is an important concept, particularly at its initiation. But the exact cell types and pathways remain elusive. We know about ischemic cardiomyopathy. Gillian is talking about that. That's a major interest of our group. What I'm going to talk to you about is non-ischemic cardiomyopathy. So this is heart failure with a reduced ejection fraction. And idiopathic cases that we don't know the reason, make up in some series up to 50%. The leading risk factor is hypertension, which is going to come up again and again in this talk. And with that, another risk factor that's hard to nail down is viral infection, which is also a major focus of our group. There are other risk factors that you've heard about, but I'll be focusing on these two. I'm also interested in heart failure with a preserved ejection fraction, an entity for which we have no treatment modalities, even though they account for half of all heart failure hospitalizations. Hypertension, again, is a leading risk factor. There are many risk factors, but hypertension is the leading one. And as you've heard, the development of heart failure is thought to be multifactorial. So we have risk factors, you have environmental factors and genetic factors that all come together. I'm going to focus in on immune cells and the innate immune macrophage as an unappreciated central player in both hypertensive heart disease and viral myocarditis, where these two entities can come together and amplify the development of heart failure with a preserved ejection fraction, and that could also evolve into LV dysfunction. So hypertensive heart disease. So we know the pathological mechanisms in general: cardiomyocyte hypertrophy, tissue fibrosis, impaired diastolic function that can lead to impaired systolic function. Traditional risk factors account for only 50% of the risk of heart failure development, and it's unclear what promotes susceptibility. In the lab, we've extensively used a model of hypertensive heart disease that relies on subcutaneous pumps and infusion of angiotensin II. We all know angiotensin II as the dominant moiety of the renin-angiotensin system. There's also other surgical models which we haven't used, so I'll be talking about angiotensin II-induced hypertension. We also are heavily invested in viral cardiomyopathy. Viral infection has long been implicated and predicted as a direct cause of non-ischemic cardiomyopathy, but the true incidence and prevalence is difficult to determine. In some series, patients who have myocarditis have very high viral genomes, so 67% of patients in a particular study versus 11% of controls, but there are a lot of controls, so there's really large numbers of patients with detectable viral genomes within the myocardium, but they don't develop heart failure, or at least overt heart failure. Animal models, there's two main viruses that are used. There's Coxsackie virus, which is the most common. It's also a human pathogen. And we use a similar virus called EMCVD. It's a cardiotropic virus. And what we want to do in general with viral cardiomyopathy is to use contemporary immunological techniques to sort out the cell types involved. So I'll just give a brief description about the research themes in our group, and it really focuses in on the role of immune cell diversity in heart failure. So Gillian did a nice job, so I don't have to delve into too many details, that the macrophage is more than one cell type at the transcriptional and functional level. So we have embryonic-derived macrophages. Even that, there's diversity. Some come from the extra-embryonic yolk sac, very primitive. Some come from later in development from fetal monocytes. These possess many regenerative, reparative activities, which she discussed nicely. Adult-derived macrophages we try to categorize them in a simple way. They promote inflammation, they promote tissue damage, and one of the last questions from Michelle was focused on plasticity, and they are a plastic cell, and they can adopt a reparative phenotype. And we study these cell types in ischemic injury, myocarditis, hypertensive injury, and more recently, how macrophages intertwine with cell-based therapies and bioengineering techniques. We think this is a critical factor in these types of therapies that should be included, as the cell type serves a role of producing growth factors for the cells that we want to reimplant. And ultimately, we want to assess the role of each individual macrophage subset in the development of heart failure. So what do the embryonic macrophages do? What do the yolk sac macrophages do? How are they different from fetal monocytes? And with that, we have human studies ongoing with cardiac tissue, where we try to relate the subsets of macrophages we see in the mouse to human, and we do see, as Jillian pointed out, similar macrophages at the cell surface marker level and at the global transcriptional level. And a particular interest of ours is to try and define a macrophage composition and gene expression patterns and how they relate to clinical indices and outcomes in patients with heart failure. Today, I'm going to focus on the interaction, though, between hypertensive heart disease and viral myocarditis. So, a little bit more background, but I'm going to go quickly through this. I'm the last speaker. I'm sure everyone's tired. During development, we have the embryonic macrophages that seed all developing tissues, and they proliferate to expand their numbers. And we have shown in the neonatal heart, when it's injured, it can regenerate. And what it does is it expands its neonatal embryonic pool of macrophages. It does not recruit any adults or circulating cells, and it's able to fully regenerate. And if you knock out the macrophages that are present, you lose the regenerative ability of the neonatal heart or other primitive organisms like salamanders. Adult heart retains, for the life of the animal, its embryonic macrophage population, but it's joined by cells from the bone marrow that also reside within the heart. We use a lot of tools. Flow cytometry allows us to mark cells by their temporal expression of cell surface markers, but we also use more permanent genetic fate mapping techniques. So we cleave DNA in the cells to mark them with fluorescent proteins, either in a constitutive or inducible fashion, and other more elaborate techniques such as parabiosis and the mixing of all these together. Now, we've shown in the adult heart that it responds very differently than the neonate. The adult heart actually loses its embryonic populations in the setting of injury, many injuries, and what happens is you have a huge influx of monocytes and neutrophils, and we all know the adult doesn't regenerate, it forms scar. So hypertension and viral myocarditis. So what I'm going to talk to you about today is the preliminary data that we have for the grant, the models that we use, the dynamics of the macrophage populations in these systems, and the questions we want to answer. So hypertensive heart disease. So if you infuse angiotensin II into a mouse for a few weeks, you'll see classic changes of hypertrophy and fibrosis. So here's low dose or high dose Ang2. You can see the heart weight to body weight ratio increases as a measure of cardiac hypertrophy. Similarly, in a dose-dependent response, you see perivascular fibrosis that can extend into interstitial fibrosis. And this is where it's very exciting for us because we'll get to work with Boris, and he's really an expert in fibroblasts and their transition to myofibroblasts and the wonderful tools that he has to explore this, which we think was a central problem in what we're studying. So what happens if you deplete macrophages in the setting of hypertensive stress? So in low-dose Ang2 infusion, you have decreased fibrosis, hypertrophy, and inflammation. And if you look at an M-mode echo image of a mouse heart, you see there's normal systolic function. Now, in high-dose Ang2, if you deplete macrophages, you have the development of severe non-ischemic cardiomyopathy. You have neutrophils that infiltrate, inflammation, and death. Viral myocarditis. So we use this model of EMCV. It's a cardiotropic virus that in the mice that we use, at dose that we use, typically they survive and they look fairly normal. The virus is cleared pretty rapidly, but even in wild-type animals here, you can see fibrosis in red as a percentage. We have a number of immunodeficient strains where this is enhanced, but even in a normal mouse, an seemingly innocuous infection can lead to subtle cardiac injury. And what happens with that? We can look at echocardiographic images. Here's a wild-type mouse infected with virus, and here's one of our immunodeficient mice. You can see the wild-type mouse looks pretty normal. The immunodeficient mice have severe contractility abnormalities in the focal pattern, and we can graph that with a wall motion index score. But in the end, wild-type animals have, they have a little bit of injury. They have subtle injury as well. And what happens if you deplete macrophages in this subtle injury system? Mice die. A proportion of them die, and they have heart dysfunction and worsening fibrosis. So even in the setting of subtle injury, macrophages are required in viromyocarditis. But the roles of the individual macrophage subsets are not known. So this is what's important. Different forms of stress elicit different macrophage responses. So this is our genetic fate mapping approach, and we're looking at flow cytometric data. Each cell is a dot. Here, all macrophages in the heart are parsed if they come from embryonic development or from the adult. That's the baseline. If you infuse angiotensin II, both subsets increase in number numerically, and the embryonic subset will proliferate and greatly increase its numbers. Now, this is very different from ischemic injury, which Jillian mentioned, where the embryonic-derived macrophages are lost, and also very different from viromyocarditis, where the embryonic macrophages are lost as well. And what's important here is this is the subset that's reparative. So here's the study rationale. Hypertension and viral infection predisposed to heart failure through cardiac macrophages. So in the steady state, we have macrophages that live within the heart. If a patient were to become hypertensive, you would have an expansion of macrophages, tissue fibrosis and hypertrophy, and there would be more of these embryonic-derived macrophages present. Now, if they were to get an innocuous infection that would otherwise be clinically not relevant, typically they would clear the infection, there would be some inflammation, there might be some mild injury. But if you put these two together, we feel that the embryonic macrophage is critical. It's critical because if you get rid of it, the heart may not be able to withstand the hypertensive stress. So in data that I haven't shown you, if you eliminate the adult macrophages, the hearts can withstand hypertensive stress, but if you eliminate all macrophages, it can't. So our thought is it's these embryonic macrophages that expand in number that are critical to prevent the development of non-ischemic cardiomyopathy. So questions to answer. Does prior resolved myocarditis predispose to heart failure development during chronic hypertension? So very simply, we will infect mice, allow them to heal, and then expose them to chronic hypertension and look at a number of outcomes, echocardiographic indices, diastolic indices, and histopathological changes. And we can take the reverse of that. Does chronic hypertension predispose to an amplified tissue injury response to subtle infection? So recall, viral infection eliminates the embryonic-derived macrophages. So here, you can imagine a patient is hypertensive and gets a viral infection where the embryonic-derived macrophages are wiped out, those that have been expanded. And what effect does this have on the actual antiviral response? We've developed a number of tools to help us study that in the mouse. Lastly, we want to identify the immune cell subsets that are required for the transition, which we believe is the transition, this would be year 2, from heart failure with preserved ejection fraction to non-ischemic cardiomyopathy. And Jillian elegantly showed what she's done, where we use a tamoxifen-inducible system to simultaneously label with a fluorescent marker macrophages and also express the diphtheria toxin receptor for deletion. And we can delete those macrophages, leave untouched the other macrophages in circulating cells, and test. For example, if you just expose a mouse to hypertension and you just knock out the embryonic macrophages, is that enough to tip it over the edge and lead to systolic dysfunction? So I'll end here with the summary. So we have hypertension, we have subtle viral infection, and we have a third category, which I've defined as the magnitude of the stress or a genetic susceptibility. They're similar in a way. And if each one of these alone makes you susceptible to heart failure. Now, if you start to intertwine these, you can get development of heart failure with a preserved ejection fraction, which can then degenerate into a non-ischemic cardiomyopathy. And we think that the embryonic-derived macrophages prevent or help prevent this transition. So I'll just end there with some acknowledgments. Members of my lab that study these complicated systems and stay late in the lab, I appreciate it. Collaborators, so Boris. So we're already planning joint lab meetings together. Again, this would not have happened if it was not for the funding that we've received. And Tanya Watts, an immunologist, that will help us. Mansour, who's been a great mentor since my arrival, as have others locally, collaborators at WashU, and funding, of course, from the Rogers Center and other sources. So thank you very much. Happy to take any questions.
Dinesh.
Slava, that's nice work again. I'm just trying to understand sort of the clinical application of your work. The general population in which we see myocarditis in clinical practice, usually a young patient in their 20s or in their 30s, usually not hypertensive. And they probably have 20, 30 years prior to them developing essential hypertension. With your models, you're causing some form of injury, and you're exposing them to hypertension pretty quickly afterwards. And also the other thing that I noticed is that you have a significant amount of wall motion abnormality in the inferior lateral wall in your models. And we don't tend to see that degree of abnormality in the clinical population. They tend to have some epicardial enhancement. In many of the times, our echoes are reported as normal. We don't even find a wall motion abnormality. So just trying to understand sort of how this translates.
So mouse to human and variability. So in terms of how you view the patient. So I would view this as someone in her 20s not developing myocarditis, but someone in her 20s developing a cold. And they didn't know it, but now they have some tissue fibrosis. And if you did an echo on them, you might see some subtle wall motion abnormalities that are going to persist for that person. They're not going to resolve. And then later, 20 years later, and you're right, maybe we should do those studies, give it quite a significant amount of time, but at least for these proof of principle studies, we'll keep the time frame fairly narrow, give them a month or 6 weeks to recover. Later in life, when they get diabetes or hypertension, we're using hypertension here, when they have a second stressor, they're predisposed. So it's not simultaneous. It might be simultaneous in the setting of an elderly person, who's hypertensive and then gets a cold 2 weeks back, and if he wasn't hypertensive, maybe he wouldn't have developed this non-ischemic cardiomyopathy. In terms of the regionality, we have mice that develop a dilated picture. We have mice where the inferior wall, infralateral wall is not involved. It's the anterior wall. It's just random. It really associates with where the infiltrate of cells is. Sometimes it's focal, sometimes it's diffuse. Sometimes it's cleared so rapidly that there is none. It is wild-type animals. So the animal that I showed you was immunodeficient. So the other one that I showed you was normal. And that's kind of the population you described.
It's interesting that you see a wall motion abnormalities in the inferior lateral wall, just because, as you know, from parvovirus B19, which is perhaps commonly seen in patients, it's been described that the most common area of wall motion abnormality is the inferior lateral wall, and it's been attributed to differences in HLA subtypes in different parts of the myocardium. So that's sort of why I was curious about the wall motion abnormality in that territory as well.
Scott.
Great talk. My question relates to the use of Ang2 as your hypertensive stimulus, and I guess, are you concerned that you may see neurohormonal effects, effects on fibrosis, other things that aren't necessarily connected to hypertension per se, and using the TAC or aortic banding model as a complement to what you're doing, I think would be a great idea.
Yeah, that's very fair and a good idea. That's something we plan to do if this works. Ang2 can stimulate its own cells in the absence of hypertension. So yeah, very good point.
What does Ang2 do to the immune cell makeup of the heart?
So in a very rapid fashion, you have a dramatic infiltrate of neutrophils followed by macrophages. There are all sorts of different subtypes with cell surface markers. They change quite a bit in the acute setting, and then they stabilize over a period of time to an elevated level.
But is that attributable to the hypertension pressure overload or to the direct
it's similar,
angiotensin
so
effect?
when we have done limited TAC, but when we've done it, it's similar.
Okay.
It's similar in almost every system in the mouse. You initially see neutrophils, and then you see monocytes, and they come in a similar fashion. The only thing that's different about TAC and Ang2 compared to other models where cardiomyocytes are gravely injured and die is you don't lose the embryonic population. It's the opposite. They expand in number.
And just for clarity, coming back to Dinesh's question, Dinesh, those patients that we see that appear clinically have acute severe viral myocarditis. And clarify, that's not what you're creating. You can create that in the lab if you want to create viral myocarditis. I'm assuming you're using low titer, low injury. You don't want to create acute severe viral myocarditis.
Exactly, and that's why I put up the immunodeficient. So, we can throw in a component of genetic, that we have mice that are, genetically altered, and humans have some of these SNPs, and there are patients that have these genetic abnormalities. But what we're using is a subtle infection, an infection where you wouldn't know the mouse was infected, even if you did the echo.
But your question's an excellent one and made me recognize my immunological ignorance. Why is it that all the cases of severe viral myocarditis we see are young people?
Because they have a robust, those who have the most severest fulminant myocarditis can recover as well.
It's an immunological. Really?
It's also more common than we think in the elderly people, but
Yeah.
we just assume it's something else.
Yeah, I think you're right, Lynn. That's something interesting. I mean, maybe you should have a little bit of a sub-study, because, in our unit, we get them transferred from all over the province. Like, we're the real referral center for all the acute severe myocarditis. Something to think about.
Yeah, I have a Peter Monk grant in on that, so we'll see what happens.
Okay.